Kami ucapkan selamat datang Menteri Pendidikan Tinggi, Sains dan Teknologi Republik Indonesia, Profesor Yuliarto PhD beserta rombongan, dan penerima Nobel 2022 di bidang kimia, Profesor Morten P Meldal di Universitas Hasanuddin. Assalamualaikum warahmatullahi wabarakatuh. Very good morning to all of us, Excellencies, distinguished guests, participants, ladies and gentlemen. Our warmest greetings to beloved students from various high schools in Makassar. Today, in celebration with the International Peace Foundation and the Bridge Program, Hasanuddin University is honored to host the Nobel Laureate Lecture featuring Professor Morten P Meldal, the 2022 Nobel Prize Laureate in Chemistry. Our theme today, Chemistry for a Sustainable World, Everything is Chemistry and How That Influences Our Choices, invites us to look at the world through a different lens, one that connects the precision of science with the responsibility of our daily decisions. Joining with us today, inspiring figures in the field of science and education, Professor Morten P Meldal, our keynote speaker, the 2022 Nobel Laureate for Chemistry, Professor Brian Yuliarto, ST PhD, Minister of Higher Education, Science and Technology of the Republic of Indonesia, and Professor Dr. Insinyur Jamaluddin Jompa, MSc, Rector of Hasanuddin University. My name is Arunisa and I am delighted to guide you through today's prestigious event. Excellencies, distinguished guests, participants, ladies and gentlemen, may I invite you all to stand please to sing the national anthem Indonesia Raya. Menyajikan lagu kebangsaan Indonesia Raya, hadirin dimohon berdiri. Be seated. Hadirin disilahkan duduk kembali. Ladies and gentlemen, before we proceed to our next agenda, let us watch another special performance by Unhas Student Choir. Please enjoy Paduan Suara Mahasiswa Universitas Hasanuddin.
Hasanuddin University shapes ideas into impact and leads with innovation. As the pride of Eastern Indonesia, we believe in world-class education, groundbreaking research, and meaningful community service that connect local wisdom to global progress. Today, Unhas has grown into 18 faculties and a postgraduate school with more than 200 study programs and campuses that extend across South Sulawesi and beyond. Supported by more than 3,100 lecturers, including 472 professors, the highest number in Indonesia, Unhas continues to strengthen its role as a hub of knowledge and innovation. Founded in 1956, Unhas was built on the vision to be a center of excellence in science, technology, arts, and culture, inspired by Indonesia's maritime character. Vision, we continue our mission to create innovative learning, impactful research, and community service that benefits the nation and the world. Beyond academics, Hasanuddin University offers at least 40 student activity units spanning sports, arts, science, and other specialized fields. Through these platforms, Unhas students have showcased their talents and earned countless achievements both individually and collectively at local, national, and international stages. To enhance the learning experience, Unhas also provides world-class facilities supported by advanced technology. These include the Unhas Library, Unhas Hospital, Dental Hospital, an educational animal hospital, a marine station, science technopark and fishing industry, breeding center, Rose House and Animal Center, along with the Unhas Hotel and Convention. Unhas has long embraced the Green Campus concept, preserving wide green spaces and serving as the city's forest, an important contribution in reducing global warming. This commitment has been recognized globally with achievements such as the UI Green Metric World University ranking and the title of the most innovative university in Indonesia, ranked 14 nationally and 109 worldwide in 2022. Innovation drives us forward. Unhas has launched new breakthrough as the next steps on the journey to becoming an agile and adaptable university, continuing to deliver solutions to global impact. With world-class facilities, a commitment to sustainability and partnerships with more than 450 institutions worldwide, Hasanuddin University stands as a modern, autonomous, and globally connected university.
At Hasanuddin University, every achievement is built together. Through the dedication of our academic community, with a deep contribution to education and humanity, we continue to embrace innovation and grow through science and technology. From serving our local community to contributing to global progress, we move forward into courage and purpose. Hasanuddin University, stronger together.
This is Hasanuddin University. where knowledge meets innovation, where future leaders are nurtured, and where collaboration builds a brighter tomorrow.
Excellencies, distinguished guests, participants, ladies and gentlemen, we shall begin our first agenda, welcoming remarks by Rector of Hasanuddin University. Ladies and gentlemen, please welcome Professor Jamaluddin Jompa.
Cek sound. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And very good morning. Your Excellency, Ministry of Higher Education, Science and Technology, Prof Brian Yuliarto, thanks a lot for still being able to attend this event. Although I know that you have to take very late flight. Terima kasih. Tepuk tangan buat Pak Menteri dan Ibu yang sebenarnya hampir tidak datang gara-gara pertemuan dengan Bapak Presiden. Terpaksa beliau harus naik pesawat jam 2 subuh. Ini luar biasa komitmen beliau untuk hadir. Thank you so much. Esteemed, distinguished, prominent Nobel Laureate in Chemistry in 2022, Prof Morten Meldal from University of Copenhagen, Denmark, and also Ms. Meldal, Dr. Federia Maria Hilaire. I hope I pronounce your name well. Thanks so much for making your visit. This is a very long journey, I know, but I hope you enjoy your visit to Makassar. Prof Brian, Pak Menteri, this is actually Prof Meldal's first visit to Indonesia, and he came to the right place, to Makassar. You are in the center Indonesia. Also, leaders and members of Board of Trustees, Ketua MWA, also Senate Academic, all professors, from Hasanuddin University, and also distinguished rectors, many of rectors here coming, also leaders from many universities across Indonesia, esteemed researchers, scientists, and also honorary guests, Prof Ahmad Loka. Terima kasih Prof sudah datang. Tepuk tangan dulu Prof Ahmad Loka, ini adalah mantan rektor UTB. Prof Edi, terima kasih sudah datang. And also our beloved students, both university students from many university. Pak Menteri, ini bukan hanya dari Unhas tapi juga beberapa universitas. And also we have a young friends from senior high school. Tepuk tangan buat anak-anak kita, 10 best students from each high school from Makassar. Ladies and gentlemen, it is indeed a great honor and profound pleasure for me on behalf of the academic community of Hasanuddin University to welcome you all to this very special occasion. Today, we are really privileged to host a lecture by a truly outstanding scientist, a Nobel Laureate in Chemistry 2022, Professor Morten Meldal. Please again give a round of applause for very nice visit. We are also deeply grateful to the presence of Your Excellency, the Minister of Higher Education, Science and Technology, whose commitment to advancing science, research and innovation continue to shape the future of our nation. Your presence today signifies the strong support from the government of Indonesia for scientific excellence in global academic cooperation. Actually, Prof Brian is not only a minister but also one of the top Indonesian or international scientists. Tepuk tangan buat Pak Menteri kita, bukan hanya menteri tapi juga ilmuwan. His age index actually much higher than the average of all rectors here or other scientists here. So indeed, he is a prominent scientist from Indonesia. We also honor, of course, to the attendance of all higher education leaders from universities across Indonesia, and also will be shared through Unhas TV later on. Fellow researchers, educators, students, and especially our high school students. You are the future scientists, innovators, and leaders of this country. Your presence reflects the shared belief that science is not only for laboratory and journals, but also for society, and at large, this is for all Indonesian. Anak-anakku sekalian dari sekolah SMA, kami semua menitip masa depan Indonesia di tangan kalian, dan tanpa penguasaan iptek Indonesia pasti ketinggalan dan tidak mampu berkompetisi. Kita harus menjadi negara kuat. Kalau kita tidak memiliki ilmuwan yang kuat, maka kita akan menjadi negara lemah. Oleh karena itu, kalian harus bercita-cita, Pak Menteri ya, minimal bercita-cita nanti menjadi Nobel Laureate, peraih Nobel. Nanti dilihat bagaimana presentasi Nobel Laureate. Ini yang anak SMA yang mana ini ya, nggak jelas mana SMA, mana mahasiswa ini. Boleh angkat tangannya anak SMA. Oh, kumpul di sana Pak Menteri. Sebelah kiri sana anak-anak kita yang dari SMA di Makassar. Tepuk tangan buat anak-anak SMA. Ya, ya. Kayaknya ada anak saya juga di situ kalau saya nggak salah ini. Saya belum cek. Ladies and gentlemen, today's lecture carries a powerful and timely theme, chemistry for sustainable world. Prof Meldal, I don't know, everything is chemistry. Yeah, I think that's the kind of the scientists from chemistry always say. Everything is chemistry. But the topic also improve how they influence our choices. These themes remind us that chemistry is not an abstract discipline confined in a textbook or research facility. Chemistry is a present in the air we breathe, water we drink, the food that we consume, the materials, all of the materials here we see that we design, and also technology that we rely upon. Every choice we make, of course, we were really related to chemistry. So how we produce energy, how we manage waste, how we develop medicine, and how we protect our environment is one way or another is chemical choices. Actually, I remember one day when I was still high school, seperti anak-anakku sekalian, waktu saya SMA, someone told me that falling in love also indeed a chemical process. I'm not sure if this is true, but he said yes. Fall in love driven by neurotransmitters like dopamine, norepinephrine, and oxytocin, something like that. This chemical create intense feeling of attraction and euphoria, often lowering serotonin levels, which can lead to obsession, anxiety, and so on. So from that time, I really in love with chemistry, although I become marine scientist, but I still really admire chemistry. Professor Morten P Meldal's groundbreaking work, particularly in the developing of what we call click chemistry, has transformed the way scientists think about efficiency, precision, and sustainability in chemical reaction. His contributions demonstrate how fundamental science can lead to a particular solution with profound impacts on medicine, material science, and sustainable technology. For our students and young learners, today's lecture is more than an academic event. It is an invitation to be curious, to think critically, and to understand that science carries responsibility. This choice you will make in the future as a future scientist, policymakers, educators, or informed citizens will influence the sustainability of our world, including our country. For our researchers and academic leaders, this gathering here as a reminder the importance of collaboration across disciplines, institutions, and also nations. Global challenges such as climate change, environmental degradation, and health crisis cannot be solved in isolation. They require science which is open, ethical, and also guided by vision of the common good. Ladies and gentlemen, let us take this opportunity not only to learn from Nobel laureate, but also to reflect our own shared responsibility to ensure that chemistry, science as a whole, serve humanity and the planet in sustainable and responsible and equitable way. Professor Meldal, it is a great honor again to have you with us today. We look forward to your insight, your inspiration, and also your wisdom. Mr. Minister, we also ask your advice and guidance and insight for us to move forward in improving our scientific culture of excellence, especially to eastern part of Indonesia, because many of the participants here mostly from eastern part of Indonesia. We need a better networks, collaboration among Indonesian scientists, but also with prominent world scientists like Prof Meldal. Di sore ini Pak Menteri, nanti kita ada diskusi mendalam dengan para ilmuwan dari universitas lain, boleh juga ikut. Mohon perkenan Pak Menteri nanti juga memberikan arahan. Lastly, I would like to appreciate and thanks everyone, especially for organizing committee, also to the Ministry of Higher Education and Science Technology, and also sponsor from International Peace Foundation, and of course for all participants for making this event successful. Thank you all. Enjoy the lecture. Wassalamualaikum warahmatullahi wabarakatuh. Good.
Thank you very much, Professor Jamaluddin Jompa, for your warm welcome to all audience who are attending today's lecture in Baruga A.P Pettarani. Ladies and gentlemen, now we will hear opening remarks by the Minister of Higher Education, Science and Technology of the Republic Indonesia. We cordially invite His Excellency Professor Brian Yuliarto, PhD, to deliver his speech on the podium and officially open today's lecture. Please give a round of applause.
Assalamualaikum warahmatullahi wabarakatuh. Good morning and greetings to us all. Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Distinguished guests, respected colleagues, ladies and gentlemen, and dear students. First of all, allow me to express our gratitude to Almighty God for granting us the opportunity to gather today at Universitas Hasanuddin. I would like to extend my warmest greetings, yang saya hormati, of course, Mr. Professor Morten Meldal, a Nobel Laureate for Chemistry, head of the Center for Evolutionary Biology, University of Copenhagen, Denmark, and Madam, of course, and then Rektor Universitas Hasanuddin, Bapak Profesor Jamaluddin Jompa, terima kasih, pimpinan Universitas Hasanuddin, Ketua MWA, serta tamu undangan lainnya, Ketua Senat. Today is, of course, a special occasion. It is great honor for Indonesia, especially for Unhas and the scientific community in the eastern of Indonesia, to welcome Professor Morten Meldal, Nobel Prize laureate in chemistry, whose groundbreaking contribution have advanced the field of chemistry sciences and enable innovations with real impact on medicine, materials, and biotechnology. Ladies and gentlemen, Indonesia is a nation with immense potential. We have rich biodiversity, growing research ecosystem, and the important one is a young population eager to learn and innovate. But to fully realize this potential, we must strengthen what I call the bridge between knowledge and impact, between laboratories and society, between fundamental discovery and real-world application. So that is why today's public lecture is more than an academic event. It is a strategic moment for inspiration, exchange, and collaboration. It reminds us that scientific excellence is built through curiosity and rigorous thinking, strong research culture and integrity, and also global collaboration. In this spirit, we hope this visit will open doors to deeper cooperation through joint research, co-supervision of students, visiting scholars program, and collaborative publications, especially in the areas where Indonesia has relevance and urgency, like in health field, environment, energy, and advanced materials. To our students and also for early career researchers, lecturers, let today be a reminder that world-class science is not reserved for a few countries. It is possible anywhere when there is commitment, mentorship, and a community that values excellence. Ask questions, be bold in your ideas, strengthen your fundamentals, build networks, and most importantly, use your knowledge to serve society. And of course, for Professor Meldal, on behalf of the government of Indonesia, I thank you for your presence, your generosity in sharing knowledge, and your contribution to inspiring the next generation of Indonesia scientists. We are truly honored to welcome you to Makassar and to the University of Hasanuddin. I would like also to express appreciation to organizing committee and all partners who made this event possible. And ladies and gentlemen, with great respect, I hereby declare that public lecture by Nobel Laureate Professor Morten Meldal officially open. Thank you very much.
Thank you, Minister Brian. Please remain on stage, and we kindly invite Professor Morten and Professor Jabaluddin Jopa to come up to the stage as well and present a token of appreciation from Hasanuddin University. We have prepared Unhas specialty as a gift presented only to our valuable guest. First, we give from Hasanuddin University to Professor Morten. And our second token of appreciation goes to Ministry of Higher Education, Science and Technology. Please remain on stage as we will take a photo. Now we request Your Honor to come down from the stage, and we invite all the participants here to stand up for a collective group photo taken from the stage. Kami silakan kepada seluruh peserta untuk berdiri dan melakukan sesi foto bersama, dan kami mengundang rekan-rekan media untuk dapat mengambil foto dari atas panggung.
Yeah, just a second.
Okay, smile for the camera. Raise your hand. 1, 2, 3. Again, give your best smile. 1, 2, 3. Thank you very much. You may be seated. Kami persilakan untuk duduk kembali. Ladies and gentlemen, our distinguished VIP guests are going to have a close discussion session. Our protocol team will now escort you from your seat, and we do appreciate your presence during this opening session. To our students and general participants, please remain in your seats as we are going to continue our agenda. Kepada seluruh peserta baik dari Unhas maupun umum juga siswa sekolah untuk tetap berada di tempat. A quick reminder to all participants that today we will have a dedicated session for questions and answers. We encourage you to pay attention and note down any thoughts and inquiries that arise during the lecture. Also, for those asking about participation, do remember that certificates will be available after we finish today's session. Kami ingatkan kepada seluruh peserta bahwa setelah presentasi akan ada sesi diskusi dan tanya jawab. Silakan menyimak materi dengan seksama dan siapkan pertanyaan terbaik Anda. Sertifikat peserta akan diberikan setelah seluruh rangkaian acara selesai. Ladies and gentlemen, we shall move to the main agenda of our event today. For the students here today, this is such a special chance and worth bragging about, as you will hear from someone who has reached the very top of the scientific world. Adik-adik SMA, ini adalah kesempatan yang sangat luar biasa untuk menyaksikan dan mendengarkan secara langsung kuliah umum oleh ilmuwan penerima penghargaan paling bergengsi di bidang ilmu pengetahuan, yaitu Nobel Prize. Our guest of honor today is Professor Morten Meldal. head of the Center for Evolutionary Biology in University of Copenhagen, Denmark, who was awarded the world's highest scientific honor, the 2022 Nobel Prize in Chemistry for his discovery of the click chemistry reaction. To guide us through today's lecture and the following discussion, I am pleased to invite our moderator, Hasnawati Saleh, PhD. The floor is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. His Excellency, distinguished guests, esteemed colleagues, students, ladies and gentlemen. Professor Morten P Meldal was born in Copenhagen and earned his PhD from the Technical University of Denmark in 1986. He is a Danish professor of chemistry at the University of Copenhagen. He is the head of Center of Evolutionary Chemical Biology. He has also served the deputy head of Department for Education at the University of Copenhagen Chemistry Department. Professor Meldal is a prolific scientist. He has published more than 300 publications and holds 21 patents. Wow. He also has numerous international and national recognitions and large research awards. He founded the Society of Combinatorial Sciences, which is he also president. Professor Meldal is not only a prolific scientist, but also an entrepreneur. He has founded 3 startup companies. Ladies and gentlemen, in the world of synthetic chemistry, we are often taught that complexity is the hallmark of progress. We are taught that building the molecules of life, for example, medicines, materials, and catalysts, require immense energy, intricate steps, and often a high degree of unpredictability. But our guest of honor today, Professor Morten Meldal, looked at that complexity and saw a different path. He asked, what if chemistry could be as reliable and modular as snap-fit connection? While working at the Carlsberg Laboratory and the University of Copenhagen, he pioneered the copper catalyzed azide alkyne cycloaddition, a reaction so precise and efficient that it became the gold standard for what we know now as click chemistry, a method to efficiently connect molecular building blocks for applications in drug discovery and material science. This discovery was not just a new laboratory technique, it was a new philosophy of science. It allowed us to map DNA, track disease within living cells, and develop targeted cancer therapies with surgical precision. He has bridged the gap between complex organic synthesis and functional real-world applications. His discovery has impacted science and society. Professor Meldal's work moved chemistry away from the struggle of how to build a molecule and toward the goal of what that molecule can do. In 2022, the Royal Swedish Academy of Sciences awarded the Nobel Prize in Chemistry for a revolution that simplified the way we build our world. At the heart of that revolution was our distinguished speaker today, Professor Morten Meldal. Professor, it is such an honor to host you today. You have fundamentally rewritten the rules of chemical toolkit, and today we will hear his lecture, Chemistry for Sustainable World. Everything is chemistry and how that influences our choices. Please join me to welcome distinguished Professor Morten Meldal, Nobel Laureate in Chemistry 2022.
Thank you very much. Thank you. Can anyone hear me on this one? Is it on? Okay. I'll take this one off. First of all, hello, Indonesia. And thank you very much for a very nice introduction to both your Minister of Science, Technology and Higher Education, and also to Rector of the university here. I am very pleased to be in Indonesia for the first time, and it is a great honor for me to talk to you. So many young, talented students are together here in this room today. So I'm looking forward to talk to you. When you get a Nobel Prize, as I did in 2022, and I shared this Nobel Prize with Barry Sharpless and Caroline Bertozzi, two of my very good friends and colleagues, but we never worked together. It's just how it happens in this world today. You don't know exactly why, but many laboratories are working on the same area. So I was working on these new reactions for combinatorial chemistry. Sharpless was trying to simplify organic chemistry, and Caroline was trying to do chemistry on cells. So we worked in 3 different areas and all came up with a similar solution to the problems that we had today. When you have a Nobel Prize, as we did in 2022, you also are expected to have an opinion about everything. Well, I don't know about everything, but I do know that chemistry is everything. And if that's the case, if chemistry is everything, we really have to think about how we focus our education. Is it rule-based, language-driven education, or is it education into the world that is surrounding us, which is composed of chemistry? Everything we do, everything we breathe, our emotions, our hunger, our will, everything is controlled by chemical compounds that interact with biomolecules inside our body. The materials that we have, that we are producing, like polymers and so on, also chemistry, all of it. So we are surrounding ourselves in the world today with chemistry that we learn to control more and more accurately. So what is life? Life is essentially 16 atoms that comes together, and they they have the ability to form life. And these 16 atoms, of course, carbon is the most important one, because carbon is right in the middle of the periodic table. It's neutral in terms of electronegativity, and it makes bond to itself. So it's protons and neutrons surrounded by clouds of electrons that forms the molecules, and really it's the clouds of electrons that is towards the outer world of the molecules. So it's actually what the electrons do that is really important. They are shared between the atoms and make molecules, and as I said, carbon bonds to itself, which is essentially the reason that you can form larger molecules that can then go together and form life. You have functionality by polar bonds, and you have double bonds formed. And that's another prerequisite for life, is that you can form double bonds. So we cannot imagine a life made out of silicon, as you see in many cartoons. That doesn't happen. And these molecules, they go together in nanostructures. So now nanochemistry is becoming very important. They are shared, the forces are shared, and they are waves that respond to the environment. These electrons, they are clouds, but they're also wave functions, and they respond to the environment. So every single atom in the universe are in principle a little bit different. So that's the complexity of life right there for you, and it's inherent in the atoms. When the conditions are there, the atoms must create life. Chemistry is everything. It's a key to life as we know it. And there's 5 functions that are necessary to produce life. The first one is molecular recognition. Every time something happens inside of us, 2 molecules comes together and are specifically recognizing each other, just like a handshake. You know, we are 2 molecules that have to communicate. So we recognize each other, and then, if necessary, we can transform the molecules, like with enzymes. To make one molecule into another molecule, we need an enzyme to do the reaction because we need the same kind of specificity as we have in the recognition. We also have molecular signaling, and the signals have to be right. So if we have to interact with a molecule that sends a signal into the cell so that the cell will behave in the way that is most optimal for its survival. And then we have molecular transporters that transport nutrients in and out of cells. So when we eat something, we actually have transporters that takes all the nutritious stuff into our bodies. And finally, of course, replication, our DNA protein system, very important. We are mostly concerned with the first 4 of these processes. For some reason, it won't change. Yeah. My inspiration for chemistry comes from when I was very young, here about 12 years old, in my grandparents' farm, where I was out from morning to night playing in the forest. And this is the beautiful forest of my grandparents' farm. And as you can see, it's very green, and there's a lot of communication going on here. So just like in Avatar, there's communication between the trees. There's a bugs that needs to know where to go to get their food and so on. And this was very interesting for me. How does that happen? What is a communication that makes that happen? I didn't know about chemistry at the time, but I knew there was something there that was interesting, some natural science that I would like to consider. But the real interest comes here. Sorry, what is going on here? Okay. I had a beautiful video showing the fireworks that I made as a young person. That was my real inspiration. It doesn't work here, I'm sorry. So the inspiration came from making fireworks. And I could make these beautiful rockets that I could send over the ocean. And that was really, really interesting. So that actually caught my interest for chemistry in the end. When I have to hire a person for my laboratory, I of course want the person to be knowledgeable about the chemistry. But this is not the most important feature. The soft skills are much more important. So what can the actual candidate become rather than what is a candidate? And here we have soft skills like intuition, imagination, association, and so on. Being able to visualize, and not only visualize the chemistry and the pictures, but also to visualize abstractions, to know chemistry by pictures, because chemistry is a three-dimensional world. And that allows you to use your creativity and your observation to look into this three-dimensional world and find where the novelty is. Have the courage to tell your PIs that you want to do what you think is most important, and then do it. Perseverance is a very important element of science, and courage. But the road to success in science is never straight. So if your supervisor is telling you that you should do something from behind the office desk, it's very likely it won't succeed. Highly likely it won't succeed. And you then have to fail. You will fail many times in your scientific career. And then you rise again and you try it another way. And then you observe, and if you make an observation that is out of the ordinary, out of what you have expected, then you are really lucky, because here is something to work with. If nature shows you a path, you should take it. And you should use your time optimally, because the time is not enough. It's always time that is the problem. And you have to get out of your comfort zones. You will never be comfortable as a scientist. You always have to step outside your boundaries. And then you can change your field once in a while as you grow up as a scientist, because then you can bring your experiences from one field into the next field and make new discoveries. But the study never ends. I'm still studying. I'm still going to the lab almost every day. And I think it's very important, because it's in the laboratory, in the fume hood, that you make the observations that are going to give you the future. This one here, also a video that for some reason won't play here. I don't know what. There's a problem. For some reason it won't play. Maybe if I do. Anyway, so this was T-Rex. T-Rex is a dinosaur. So he is going hunting, and when he has been hunting, he is filling his stomach, so he feels relaxed. He's controlled by the aggressor molecule adrenaline, putting all the blood to his legs during the hunt, and then the small molecule noradrenaline when he is finished hunting and he wants to relax. And then when he is full, his tummy is full, he will be rewarded by dopamine and serotonin, which is a cognition molecule. So these are the simple molecules that the simple animals of 65 million years ago were controlled by. Today, we are a herd community of human beings controlling the earth, and this herd community is controlled by different types of molecules. Here you see the love molecules, oxytocin. Oxytocin is there to create empathy, to create collaboration, to create a society that is coherent because we help each other to achieve the goals optimally. This is the power of the herd, and this is in danger at the moment, as I will come back to. The other molecule you see here is the vasopressin, which is a heart pressure molecule. And this molecule is a very simple modification of the love molecule. So you can see here, nature is cheap, but if we look at the oxytocin, this is a three-dimensional structure of oxytocin. The oxytocin is really looking like this, like a patch of chewing gum with color. So this is the electron clouds that the blue and the red shows you deficiency and surplus of electron density. And this is what actually controls everything. Everything in life is controlled by the interaction of these polar bonds with a receptor shown up here in the corner. The interaction with the oxytocin receptor sends a signal to the inside of the cell through the cell membrane, and then the molecules on the inside, they go to the nuclei and they activate the nucleus for expression of a particular protein that is required to feel empathy or to feel nesting. So we have pleasure, love, empathy, and so on from these molecules. So I thought that peptide materials and peptides is a boiling point for interesting research. And that's because the proteins and the peptides have all the controlling functions in human beings, in life in general. The enzymes, the signaling molecules, the hormones, everything is made out of peptides and proteins. So this is where we can find pharmaceutical potential As you know today, one of the biggest pharmaceutical successes ever is a GLP-1, which is not only treating diabetes, but also has the cardiovascular effects and the slimming effects on people who are obese. So that's a magic bullet, and it's a peptide drug. We can do them by virtual design on computers, and we have a biology that is very active within this field. So I think that was a very exciting area. And we formed a research center at Carlsberg Laboratory where we would work with new organic quantitative chemical transformations for Copenhagen. That's a very long word, quantitative chemical transformations. Unfortunately, Sharpless's wife, Jane, came up with a better solution, and she called those reactions for click chemistry. So click chemistry is the persisting name for these kind of reactions that we were developing. And our purpose was, of course, to develop them for drugs, but they showed much higher potential. So we were making polymers that were compatible with biochemistry and also with chemistry. So we were able to synthesize compounds inside these polymer beads that you see here, and then we could test them biologically with proteins and cells and so on. So this was the foundation for combinatorial chemistry, and we have investigated a large number of enzymes like proteases, like glycotransferases and so on in these beads using a variety of peptide chemistries and organic chemistries. So we developed all of these organic chemistries for combinatorial chemistry. That means that we had to develop them for quantitative chemical transformations. And you will notice one of them is a QAC reaction, and that QAC reaction is an archetypal click reaction. So first, how do we do the polymers? We take a macromonomer mixture and we inject into this an initiator, and then we form small droplets, and these small droplets then are polymerized in a heated coil, and we can collect the beads that forms in this way. And this is a reason that we get these absolutely perfect beads that you see in this slide here. So our best beads are with very small variation in size, meaning that we can now use these beads for quantitative bioassays. They look like that, and this is just showing you the beads. And furthermore, we are able to equip these beads with a small barcode. So each little bead of 0.5 millimeter contains a barcode that we can read at any point, just like in the supermarket. When we do a reaction, we can read the reaction, what was done, and we have a matrix of information afterwards that we can use to decipher the compounds and the assays. So this is what it looks like. We have a three-dimensional matrix of small beads that are immobilized inside the polymer. And here are some examples. There are exposed red and green beads here. And they are completely overlapping, and that's because there's a week between two readings, and we recognize the same bead exactly. And there's a very high encoding potential in this methodology. So we have now a technology which is combinatorial chemistry, starting out by mixing up in the right corner. You can see we are mixing the compounds for every synthetic step. We build up millions of compounds, and then we can screen them with a biomolecule, for example, an enzyme. As you see here in this image, we have 6 small beads that are active in a very large background. We isolate them, find out what it was that interacted with the enzyme, and then we have a tool to investigate the enzyme. So that's what you see in the mouse below. The mouse has arthritis in one foot, and we have made a substrate which light up when the enzyme is active, an enzyme that is characteristic for arthritis. And that's what we see in this mouse foot. We can also grow our cells on the surface of the beads using an adhesion peptide that maintain the contact to the cells, so they grow as monolayer on the surface of the beads, and then they can interact with the substances that are sitting on the beads and give a signal. So here we are isolating G-coupled protein receptor ligands that are highly active. Down here in the lower left corner, you see an image where you have one active bead and you have a number of inactive beads, and we can take the active one and find out what was active with this GPCR. And we can use our encoding system to actually identify the structures. So this is our platform, and we were working with these kind of molecules to synthesize difficult peptides in our laboratory. And it was very successful. We could make these peptides that you couldn't make in any other way. So we thought maybe we can use these kind of molecules for other things as well. And I asked Christian Tornø, one of my PhD students, to try to do this. React an alkyne, first form the anion of the alkyne as a copper acetylide, and then couple that with a very reactive acid chloride with an acido group on the other end. That would be a perfect substrate for making something like GLP-1, where you have this fatty acid chain sticking out from the middle of the peptide using a Diels-Alder reaction. But he couldn't do it. He tried and he tried and he tried. This was one of those cases where your supervisor tells you something and it totally fails. Okay, so my idea of doing this was not working very well. We tried and tried for months, but in the end we had to give up and just look at what happened instead. And what happened was that we formed this compound here, still with the acid chloride intact, which was very unexpected. So we followed up on this, and we actually investigated the compatibility with all biomolecules of this reaction, and it was very compatible. So Christian Tornø here on the right side, he went to the APS in 2001 and presented this result. A great result. We didn't know at the time that this would be so important, but we could see what happened. And what is then the click chemistry? Well, here's an alkyne. It's like a bag of electrons, and then from the top here, it finds a positively charged copper atom. The copper atom interacts with the electron cloud and acidify the alkyne, so the proton falls off and the copper takes its place. Not much happened by that, but then a second copper atom comes in here, and it sits on the electron clouds and strips off the density from the alkyne. Now we have an empty room on the alkyne. It needs electron density. Where does it get it from? Well, out here from the corner. First you get these empty orbitals that you see here, and then out here from the corner comes a very electron-rich compound, which is the A side. And as you can see, the A side fits almost exactly in terms of how the orbitals are arranged and their faces to make new bonds. And it does so, first making a metallocycle that spits out one copper atom, and then we protonate the other one and get rid of that copper atom, and Christian was very happy. But if you ask, is this an important result? We have coupled a phenylalanine with a methyl group. You might say, that's not very important. But the fact is that this can be used for so many different things, and that's where we have made chemistry simple. The only caveat of this reaction is the potential for oxidation of copper 1 to copper 2, which is very high, so you cannot carry this out in the presence of oxygen. That's the only caveat of this reaction. Because if you do, you get reactive oxygen species. You all know we have to avoid radicals in our diet. And these are the radicals that we are going to avoid, because as you can see, when we treat in air a protein with copper 1, in this redox cycle, you will see a lot of oxidation of the protein down here in the lower right corner. we see addition of 16, 32, 48, and so on. So a lot of oxidation is going on, and we have very little of our original protein left. So we have to keep away oxygen, and we have to stabilize our copper-1 catalyst. If we look at a click reaction, what is the minimal criteria for a good click reaction? Well, we need to have a high driving force for the reaction, right? So we need high enthalpy to make it quantitative. We also need to be orthogonal. That means that we can do the chemistry in the presence of other functionalities. And that's why we need orthogonal functionalities that are easy to install on our molecules, so we can build any kind of structure. We need it to be biocompatible and clean at 20 degrees, stereo and radio selective, meaning that we have total control of our chemistry. It should be insensitive to general things that are in the air, like carbon dioxide, water, and oxygen. There, our compound actually fails. Our reaction fails a little bit. And then it's best if it's coordinative, because then you can go into a cellular environment and you can acquire the component, put them on a template coordinatively, and then these will react with each other very selectively. And then you need to provide otherwise inaccessible molecular architectures. So all of these things have to be fulfilled, and that's why it's so difficult to find a click reaction. Today, 26 years after the discovery, we only have 5 click reactions, 4 to 5 click reactions. And they are all very, very good, but they are very few. And I urge people to think about this, because if we have also orthogonality between different types of click reactions, there's no end to the future of chemistry. So what we do is that we inject our mixture of click components. Usually we use this for cyclization, and then we have a dilution principle in which we make the reaction happen very small portion at a time by continuous injection into a click mixture. So that works very well with copper triflate, and we get the products we want. For example, here's 3 components. There's a component that can kill a cell via interaction with a cancer inhibitor, cancer-specific inhibitor of the cell death. Then we have something that can enter the cell, and we have a location device associated with this. And if we put those 3 molecules into a container with copper-1, like this, and steer them overnight, we get a single molecule which now have 3 functions. A molecule that can recognize, can import, and can kill. So it's just like making a robot, actually. So molecular robots that can have multiple functions can be created by click chemistry, and we can do the components either by amber stop codon technology or by synthesis. So here we have this molecule. It recognizes a cancer cell by the recognition domain, and then we have a cell entry domain that is used to import the other 2 functions into the cell. And then the cell, of course, will undergo programmed cell death, because we have blocked the inhibitor of programmed cell death. So we can make molecular robots. We can also use it for ligation, for surface modification, polymer synthesis, and so on. So many different areas where this chemistry just works. And that's the reason that it has been awarded. And here we are at the award ceremony. This is a beautiful holiday, highly recommended to all of you. In Stockholm, where you are invited for a 6-day or 7-day holiday. And this is the first day where you are at the museum where you deliver something to the museum that has been very important for you. This is our first synthesizer that I made in my neighbor's garage, which is used for combinatorial chemistry. And then we, of course, also have to sign here the chair in the bar, very important thing. So here's my signature in gold under one of the chairs in the Nobel Museum. These guys, so I will now give some examples of how to use click chemistry. These guys are 400 million years old, not these ones on the picture, but as a species, and that means that they have very good protecting system. Otherwise they would have changed. They look completely as they did 400 million years ago, and they look like small tanks with wheels underneath and a lot of sensors to sense around them. The most important thing here is the blood. The blood can detect foreign organisms and engage them, and then it can inject toxins like the thing here up in the right corner over there. That's a toxin that can be administered to the trapped foreign organism, and then it will die. The way it works is by cell permeation, and this is target-placine, a small molecule shown here at the top with two disulfide bond bridges. We thought maybe we can use our click reaction to substitute these disulfide bonds in the peptide and make an active compound that would be also useful in a resistant bacteria. Because one of the problems with all these antibiotics is the multi-resistance that is currently being developed around the world. In this process, we also discovered that the way they work, these molecules, is that they form a dimer, hiding away all the unpleasant lipophilicity on the inside, and then they have a benign arginine shell, so they fly around in the serum of the body until they meet a negatively charged membrane like the bacterial membrane. And when they do that, they sort of collect themselves onto the membrane like this, and they form a pore through which the interior of the cell can escape, and when it does that, the cell will die. So this is how the Japanese horseshoe crab is getting rid of foreign organisms. So we were very interested, as I said, and we built these molecules, and here is 4 different cells that we tried this molecule with: coli, staphylococcus, salmonella, and bacillus subtilis. And in 3 of these, the natural compound which work in a physical manner actually worked quite well. But in the 4th one, there was absolutely no activity. Our compounds, which is the red and the green, on the other hand, were very reactive in all of them, and even more reactive in the subtilicin case. So here we have a story that tells us maybe we can use click chemistry to overcome some of the multi-resistance in our current administration of antibiotics. I said initially that we work with chemical recognition molecules that should actually rival the activity of antibodies. So this is our concept of an antibody, a small beta hairpin peptide. It is a strand, a turn, and another strand with a bridge of click chemistry. So the strands are maintained by the blue residues. So we know now the structure of this beta hairpin, and the red residues can be modified in the computer as we like. So we do that, we do virtual combinatorial chemistry, making mutations all the time while we are optimizing the interaction for a particular protein site. So here's an example where you have interleukin 6 and you have the 2 beta bodies that we have designed sitting on it in red and blue, and the protein is in green and orange. And when we mix the red and blue molecules together, nothing happens. And then we just add a tiny, tiny amount of the interleukin 6. And now you see the recognition first on the bead, and then we take in the molecule that is in solution with the fluorophore, and we get a fluorescence bead out of it at only 20 nanomolar concentration of the interleukin 6. Can we use this for anything? Yes, we can. This here is a plaque of Alzheimer, a plaque where we have fibrils that forms inside the brain and precipitates, and it's a very serious issue because right now 65 million people are affected by this disease. In 2050, we expect 130 million people being affected by Alzheimer. So it's a very important issue to try to remedy something that can cure this or can get us rid of the fibrils, can get us rid of the Alzheimer. And the crystal structure that you see here to the right, or that's an electron microscopy image of the fibrils by Grima at Allied, and that one we took because that one was actually isolated from fibrils, and then we cut it in two and we made these beta bodies sitting on the growth interface for making these fibrils. That worked quite well actually. As you can see here, the two to the left are fibrillation curves, and you can see 1 to 42 form these nice fibrillation curves. And when we add our molecules as in the middle, you can see that we can suppress, to some degree, can suppress the fibrillation. We can also dissolve already made fibrils to a certain extent. We were not quite happy with the results. The peptides were not very soluble and there were other things, issues with it. So Yuan Zhang, a Chinese PhD student in my lab, he synthesized this building block from something as simple as glucosaminic acid. He did the FMOC installment, the silylation and the hydrolysis to get this building block for peptide synthesis. And then we synthesized a number of peptides with these building blocks inside. Why would we do that? That is because we're now sticking out 4 hydroxyl groups from each of these residues into the water. And these are hydrogen bonding to the water, making a water shell on one side of the molecule, which is really helpful for the interaction with the fibril. And that's what you see here, the designed molecules that bind to the fibril, and they have this GA, or glucosaminic acid residues, in the peptide. And here you see the result. We can completely suppress fibrillation with these two compounds, compound 3 and compound 5, sitting on each end of the growing fibril. So that was very successful. What we imagine happens here is that we have the water shell helping the addition to the compound's surface, to the fibril surface, and then you get these kind of fragments where you have the water shell peptides sitting on small fragments of the fibrils that we can then get rid of because now they are not a precipitate but a soluble component. So that's our solution so far for Alzheimer. During this time, now we're back in Stockholm. This is a very humble moment of your life, because this is the day where you give the lecture for 10 million people. So this is nothing. You should try 10 million, that's a lot. And then after this lecture you are very relaxed because it went well, and it went well for all of us. So this is the 3 of us having a nice moment after our lectures. So Martin Wolfram was also working on Alzheimer. He was trying to make small molecules that could go in and cleave these toxic peptides, the Alzheimer A-beta. So if we could cleave them and they become small components, maybe we could get rid of them before they did any harm. And he made these peptides which bind to transition metals with carbines formed inside the peptide backbone. and then we bind that to a transition metal that can then help the catalysis. We did that carbines, we also did it with bipyridyls, like here. Not that you need to know what bipyridyls are, or carbines, but just know that we can make these kind of catalytic peptides that recognize Alzheimer peptides, and they worked fantastically. So we could actually cleave these peptides, we thought. But then Martin Wolfram, he took out half, still worked. He took out 5-fold, still worked. And he took it out completely and it still worked. So this is the second time we see something that is not expected. And this is where your research really can make a turn. Because now we have an observation that we have cleavage of the Alzheimer peptide by autocatalytic mechanism. So it's just doing it without any additive at all. So this is what we saw, the cleavage in the middle of the nucleation site for fibril formation. Perfect. And it's done all by itself in an autocatalytic fashion. So we made all the peptides that you see here, and we tried to investigate this process in more detail. And this is what we saw. You can see the red line up in the left corner there. The red line is the starting point. After 11 days, we see something else happening, and then something else, and then something else. So this is actually the pathway of cleavage of these compounds. So we now know exactly what is happening, and the nice thing, this is a shorter peptide. It's not the full length. But if we took a longer peptide and added some of the active substance here, we also get cleavage. We could even take the full length 1 to 40 and add a small amount, and within 12 days we get complete cleavage. Without addition we get nothing. Then we took 1 to 42, so really the bad guy in the Alzheimer peptide, and we left that for a while. First for 1 month nothing happens, for 2 months nothing happens, for 3.5 months, and suddenly we saw this thing down in the corner here where it's completely degraded. So it takes time. There is a latency period which is actually fitting very well with the fact that you can have Alzheimer for a number of years before it really manifests, right? So here we have a latency period that is very long for the Alzheimer peptides before they do this autocatalysis. The other thing that we observed here was that there's a mechanism for the fibrillation, which is first formation of a donut of single-stranded peptides. Then the donut collapses into strings. As you can see, there's like a key over there. This is a string that is under formation. And then the strings come together as a long train that mature as a mature fiber. So this is how we form the fibers. But more importantly, when you have Alzheimer, the neurons die. This is actually where Alzheimer is very bad. The neurons are killed by this disease. And the way we believe that that happens, or one of the ways that happens, is by hydrolysis from this peptide, aggregates of Alzheimer peptides, of the adhesion molecules that keeps the neurons together so that they can communicate, so that we can have a brain that works. So if we break those peptides or proteins that sit and hold together the neurons apart, they cannot speak with each other anymore. And of course, that's very damaging to our whole brain process. And this here is the sort of the structure of these peptides, of these proteins on the surface. You can see the cell membrane. And in the Alzheimer patient, these soluble fractions of that proteins increase dramatically. And in particular in areas where you have neuronal cell death. So there's high likelihood that this is actually something. Here you see the soluble fraction in the healthy and the non-healthy cell, and this is very different. We can also see so this is a protein, and we have co-localization of the soluble protein and the beta peptides in the brain. So it's highly likely that because of this sequence similarity between the Alzheimer peptide and this region of the NCAM2 molecule, the adhesion molecule, it's because of this similarity that we get this side effect. And that's probably the reason for neuronal cell death. So here we are, and we have this aggregate of the peptides from the Alzheimer that comes in, sits, cleaves off this part, and now the cells cannot communicate anymore. So we have a way that the fibrils are formed. We have the donuts to the fibrils. We can block the fibril formation, and we have also discovered that we have autocatalytic enzyme formation in this Alzheimer peptide. Now we are back in Stockholm, and this is a most important day because this is when you actually get your Nobel Prize. So here I say hello to the Swedish king, and I get a Nobel Prize. But that Nobel Prize is immediately taken away from me again. So, you know, I was thinking, maybe I said something wrong to the king. But the fact is that many of us, Nobel laureates, did our experiments 40, 50 years ago. And some of us has gotten Alzheimer in the meantime. So a number of the Nobel laureates has forgotten their Nobel Prize in the ceremonial hall. But I was lucky, I remembered mine, and it is here. So it was not because I remembered mine, it was because, oh, what happened here? There. Can you go back to my presentation? If I go here, slideshow. There we go. Sorry. So I said hi to the king. I got this one, and it was immediately taken away because you only get this on the last day. It's given to your family, and they bring it safely home. We are also working on transport. So this here is the master of transport in nature, the TAT peptide from the HIV. This peptide is a multi-charged peptide with a lot of arginines and lysines. Here we go. Lots of arginines and lysines. It's a bit toxic actually, because it interacts very strongly with the cell membranes. So it's still working well to import stuff into cells, but we thought maybe we can improve it by making it much shorter and only have 4 charges instead of 8 charges, because then it wouldn't be that toxic. So we thought about it, and we came up with this idea. We want to make a turn of the peptide, so a little bit of a beta hairpin, with a proline that has a lipophilic sticking into the membrane and 4 charges, and then we would make it of D amino acids instead of L amino acids, because then it would be metabolically stable. And finally, we would cyclize it to maintain a structure like this one here. And then we will see if we can get it into cells. So this is a structure of the peptide. So we needed the proline derivatives, and I won't go into a lot of details with the synthesis, just here to show you that we made N-linked and O-linked derivatives of proline. And these are then used in the construction of peptides. So we have the 4 building blocks here, 5 building blocks, and we incorporate those building blocks into the peptides. So these are the 16 peptides that we made. You cannot see the structure here, and that's not important. Just notice we have 16 peptides that we are then investigating for the import ability. And here you see the result. And if you look very carefully, you can see down in the right corner, we have on the second last position, we have some import going on. All the rest of the molecules we made were almost inactive, but this one was very active. And if we take that and blow it up, you can see it better on the top. And then we compared that with the HIV peptide, so the natural one, and it's actually much better than the natural one. We quantified it, and here you can see the results. So our peptide is really, really good at importing, here in this case, just a fluorophore into the cell. So we thought, can we also do something with proteins to get them into cells? Well, first, here's how we synthesized it. This is the peptide with the fluorophore. This is the atota on the TAT peptide, so a fluorophore on the TAT peptide, and these were used in the studies. We synthesize any kind of molecule on these small synthesizers, and we actually fabricate ourselves in the lab. They are beautiful, much better than the commercial ones, because we can do anything on them, and we are completely in control of what we are doing. So we investigated the mechanism for the import. It is temperature dependent, meaning that it is an energy dependent function. We can see that it takes 12 hours to reach the optimum conditions, the optimum import. We can also see that it works down to nanomolar scale, 100 nanomolars. We still get good import. And we could also see what kind of mechanism it's working by. It's here a endocytotic pathway, and it's dynamine dependent. From the addition of inhibitors of different pathways, we can see that this is a dynamine dependent formation of an endosome that is taken into the cell. Now there are two things that can happen. We can either release that from the endosome inside the cell, or we can fuse on the far side of the cell, which would be perfect for blood-brain barrier transport. So we can also use this peptide in any kind of cell line. We don't have any limitations there. And then we thought to ourselves, can we use this for something useful, like importing a whole protein into the cell? So we made this peptide, first clicked the peptide and then added a click functionality again. And we clicked it together with the green fluorescent protein, a fairly large protein, to see if we could import that. And the upper line here is just a green fluorescent protein. The second one is the two molecules separate. Nothing happens. We haven't clicked them together. We just put them into the cells, and we see nothing comes in. And then we click them together, and now we can see the import is amazing. We get it into endosomes, and they are transported through the cells. So this is what happens. We have this formation of the interaction with the cell membrane. It buries the hydrophobic part into the membrane and interacts with phosphates, with the arginines at the cell surface. And then it forms a vacuole that is transported into the cell. The last thing that I will show you is how we can use this also in biology. Because one of the most difficult things to express is proteases. They have proteolytic activity, meaning that they cleave proteins. So if you try to overexpress a protease in a cell, this protease will cleave the host proteins. And of course, this is not a doable thing, so we cannot have expression of active proteases in cells. So this is a TEV that we use quite a lot in the lab, made by the tobacco edge virus. It's a protease that has a very specific cleavage site. The coli are not very happy when we are trying to express this. As you can see, they are very unhappy. But we thought maybe we can just cut it in two, so we will have two inactive parts, make them separately, and then click them together again. So we did that, and we expressed them in separate containers. Here you see how they are moved over there. And now we ask the coli if that is okay. Here, are you glad now? And it says yes, it's very happy. So we could get several hundred milligrams of protein out of that, and we could combine these two. Oh yes, first of all, we did that with two kind of linkages, a long linker and a short linker, to combine the two residues. The short linker was a bit hindered. The short linker was a bit hindered, as you can see. It's not clicked quantitatively. But if we take the long linker and mix them one-to-one, we have a completely quantitative transformation into the active protease. That's the one on the lower left corner. No, lower right corner, sorry. So the long linker is the one that works. So we did that, we expressed it with a long linker, and we put them together, and we came in with a copper 1 residue that made the triazole. Now we have a protease, and we ask ourselves, is this active? We use the substrate, which is a fret substrate. That means that it's quenched when it's complete, and when we cleave it, it starts to fluoresce, and you can see the fluorescence curves here, and from those curves you can see that our protease is even a little bit better than the natural one. We are back in Stockholm. This is the last day, and we are going to the ceremonial dinner, second to last, ceremonial dinner. This is my wife. She is very proactive. And you can see that she is wearing a very artistic outfit, which is made by the arts faculty on the Stockholm University, because it was on the museum on the first day, and my wife took the telephone and said, "This is the one I'm carrying at the ceremonial dinner." And they said, "Yes, of course it is." So they made her one that fits exactly, and she is wearing that. So when you dress up your wife like this, this is what happens. Prince Philip of Sweden comes by, finds my wife, and I'm put to the side, right? I get a seat down by the door in the draft, and she is seated in the middle of the ceremonial hall, together with Prince Philip and Prince Daniel. Bummer. But that didn't really matter, because the next day we were at a private party at the King's Palace. Yeah. And there you have your partner at the table selected, and I was very lucky to get here by Victoria, the coming Queen of Sweden. You can see how happy I am by the goofy look in my face. Yes, so I would like to end out with a little bit of non-science. This here is a slide that tells you that the world is a dangerous place. It's becoming more and more dangerous every day, both due to political and to physical conditions of living here. So many people are suffering in this world today, and we should prepare for it. The preparation is called education. That's our only solution to the problems that we are facing. Political as well as global warming consequences. So we have an autocratic trend in this world, and that is because human beings, when they are threatened, has a tendency to follow a leader like lemmings. They are going towards the precipice, right? And they are just following over the precipice. So you should be very careful with what you actually think and who you actually elect, because the only way we can come through this is as a herd, as people who use oxytocin to feel a common good and to actually go for that. So we have a world that is currently ruled by narcissists engaging in senseless wars for their own benefit. We have a lot of global warming consequences that don't get any attention because of all this havoc. But they are really the threatening thing in this world. We are having ice melting, plastics, pandemics, floodings, fires, deserts that expand, loss of diversity in our biology, we have resource depletion, and so on. All of these things have some association with chemistry. And education in chemistry is our only solution to the worst challenges that has ever faced human beings. We have only seen the top of the iceberg so far. I think we should make this kind of arrangement 5 minutes videos to visualize chemistry for first graders, 10 minutes every week for second graders, 15 minutes for third graders, and so on, until we have given the students 400 hours of animation of the chemistry world. That will leave them at the level of a bachelor while they are finishing high school. I think that is necessary in order to combat all the challenges that we are facing. So we call this AI, and it says, what can I help you with? And you just give the grade and the week of the school year, and then you get the video material you need, VR and so on. And this is very good because visual material is global, and we can very easily translate whatever is on this video. And it will be helpful for everything, including environment, climate, global politics, conflicts, etc. So this is a path to peace and solutions. So I think that we should actually make a molecular building system like this one to get the young into chemistry as early as possible. A molecular building system that actually works like molecule, small magnets to simulate hydrogen bonds, shapes that would allow you to see the 3 barriers of rotation around the sigma bond, or a double bond which has a very high barrier of rotation. All of that can be incorporated into the way we construct this molecular framework. Because when we are born as children, we have no language. None of you understood anything when you came out. But you start storing images, and children have a very, very powerful ability to store images. That is why every single one of us, if we think back on our childhood, we can actually see even details of experiences from our childhood. Because we are very good at storing 3-dimensional information. That's the only language we have, and we have to make a mirror, a navigation mirror of the outside world. That is done by imaging. So I think we should utilize that in the education. This is a unique opportunity for the future. And then we can build excellence, not only excellent people, single excellent people, but really excellent teams who have a sandbox of infrastructure, of tools to investigate the molecular world and make other excellent people. So there should be freedom of thoughts and freedom of action, and this is very, very important because you cannot do the observations that you need to do, you cannot do the changes of actions you need to do breakthrough research if you are not free. So you should be allowed to do so by your PIs if you have a good idea. Tell your PIs, I want to do this and that, and they should say yes. And I think that the most important thing right now is to focus a little bit on the climate problems. So here are some suggestions for areas that I think is good to choose, like new sustainable materials, new production processes, that could be enzymes or new catalysts, energy storage, how do we handle hydrogen, how do we convert sunlight directly into hydrogen, and so on. food sources, because we are going to be more and more people, and we need to utilize our ability to make food as much as possible. New textiles for clothing, because textiles is a big problem, and also new transports. Those things are very important issues in the world today, to bring down the carbon footprint in particular. Another thing is, how do we get this freedom? Because right now, you have to follow whatever your Minister of Higher Education and Technology thinks is most appropriate to research in. But in fact, it's a researcher who knows what is most important. So you shouldn't have to make these blue sky applications for research money. Rather, as a researcher, we should allow ourselves to be evaluated all the time to see if we perform good, and then given the money to do so. Today, we don't separate applied and fundamental research. It's one big pool of investment. I think this is completely wrong, because these things are very, very different. And we should separate them in two independent pools of money, where we have decided we want to use this much for research that's secure tomorrow, and this much for turning already existing research into application. Separate these two completely, and then you get disruptive research, and you also get application. Finally, education is the highest return on investment for a country. It's not for a company, but long term, and for a country, it's been proven again and again that research and education is the way to prosperity. Here we see examples from Finland, the education system that was installed there, South Korea, investment in education, Poland's education reform, and Vietnam's focus on primary education. All of these have led to prosperity without enriching a single person or single company, but to the total society. I think this is a very important fact to actually realize. And then I would like to thank you all. First, I would like to thank my collaborators. These are my collaborators over the years. Can I get that back? Thank you. And then I would like to thank all the people from the Carlsberg Laboratory involved in the development of the click reaction, and finally the application of this click reaction in all sorts of biochemistry and so on from the University of Copenhagen, in particular here Martin Wolfram and Jan Zhang on the Alzheimer project, and Sarah Mellemgaard, Christian Kofod, Niklas Henrik Fischer, also on these protease projects. Finally, Phaedra, my wonderful wife and colleague, she is a chemist, and we have so many good discussions at home. So thank you very much, Phaedra. Give her a hand. Thank you.
Thank you so much. Boleh satu mic lagi untuk beliau? Panitia mungkin bisa membantu, kita butuh mic di situ. Thank you so much for inspiring lecture, Professor Morten Meldal. I would like to just briefly sum up, just briefly in bahasa for our high school students. So, Bapak Ibu yang saya hormati, terutama adik-adik yang hadir di sini. Tadi beliau, Profesor Meldal, telah menyampaikan bagaimana perjalanan beliau di dalam meraih hadiah Nobel. Tapi izinkan saya untuk menyimpulkan sedikit apa sih makna dari temuan penting beliau yang disebut dengan reaksi Lego dalam dunia molekul. Jadi temuan Profesor Meldal ini merupakan temuan metode di mana dua kelompok kimia tertentu yang disebut dengan azida dan alkuna akan langsung mengunci jika ada molekul yang namanya tembaga. Jadi seperti perkakas, dia akan klik seperti Lego. Semua tahu ya apa itu Lego. Jadi temuan ini mengapa begitu hebat? Karena menggabungkan molekul itu seringkali menghabiskan produk sampingan yang beracun atau reaksi yang gagal. Dengan temuan mendasar ini menciptakan revolusi di dalam kimia dan penggunaannya sangat luas. Jadi kenapa metode klik ini sangat berguna? Karena dia sangat efisien, hampir 100% molekul berhasil bergabung, sangat spesifik, ramah lingkungan, dan apa gunanya untuk kita. Jadi betul-betul temuan ini mengubah cara ilmuwan bekerja. Misalnya untuk bidang obat-obatan bisa mempercepat penemuan obat untuk kanker dan antibiotik, kemudian material baru, pemetaan sel. Tadi beliau menjelaskan salah satu contoh yang beliau paparkan adalah Alzheimer. Alzheimer itu penyakit penurunan daya ingat yang berbeda dengan pikun. Kalau misalnya Alzheimer itu, kalau orang pikun cuma nggak tahu di mana kuncinya disimpan, misalnya kunci mobil. Tapi kalau orang Alzheimer nggak tahu bagaimana cara pakai kunci itu. Kira-kira seperti itu bayangannya, dan itu belum banyak yang keberhasilan untuk obatnya. Kemudian beliau juga menekankan pentingnya untuk menjaga rasa ingin tahu, bagaimana menangani kegagalan ketika kita bereksperimen dan sebagainya. Kemudian beliau juga menekankan bagaimana pentingnya proses kimia itu. Segala kehidupan kita ini adalah kimia. Jadi saya kira itu saja. So I have just sum up your lecture. I hope I don't get it wrong. But now I would like to invite our audience to ask questions. Any burning questions for Professor Meldal? Meldal, please, panitia organizing committee, raise your hands. I saw someone there in the right hand side. Ada lagi yang ingin bertanya? Yang di atas? Waduh, itu kayaknya yang di atas harus turun. Ada nggak mic di atas? Is there any mic? Okay, there is. Okay. So one more. I would like to take 3 questions for now, and then we can continue. Okay. 1, 2. Pilih satu di atas deh. Kemudian 3. There's 3 questions now.
Stand up.
Yeah, we can stand up. It's better. Silakan.
Thank you. Thank you for Professor Meldal. It was a very inspiring public lecture. It reminds me back to 20 years ago when first time I took my chemistry class in this university. So my name is Adi Maulana. I'm from geology. I would like to ask, you know that in this current situation, we know that chemistry is everything. Chemistry shape the material, medicine, energy, and then climate solution. So can you please give me one example, the chemistry-driven change in our daily life that could have the biggest impact on sustainability if it is widely accepted by the society, and what we have to do so that change could be happen faster.
Yeah.
Thank you.
Yeah, I think that I've just given that answer to a journalist actually, because I think the most important thing right now is to figure out how we can use sunlight directly to make things that can replace fossil fuels in the processes that we have. And I believe that there is currently a lot of activity going on very successfully in converting at high percentage yield water into hydrogen using sunlight directly. So you don't need to produce electricity with solar panels and so on. You use a catalyst system that capture the sunlight energy and convert that into hydrogen molecules that can then be either converted into a storage molecule for the energy or can be handled directly into cars and so on, because that is the only completely carbon neutral technology that we can develop.
Yep.
So you
And
mean
I think the
energy.
carbon issue is the most important one right now.
Yeah.
There's other issues like how resources affect our economy and stuff like that, which makes Trump wet his bed at night. But I don't think that's the most important thing. The most important thing is to get in control of the global warming.
Okay. So what do you think would be the very big challenge to make it that kind of change happen faster?
Yeah, so the scaling is really, really important. So it must be cheap, easy, and powerful.
So it
And
should
these
be
three
economically
factors-
feasible. Yeah.
So it doesn't really help anybody if you spend 3 years to synthesize a small amount of molecules that can do this. You really have to work with scale in mind, right?
So economy is still the most important things here.
The what?
It is economically feasible also.
And economically feasible, but I think that if we take the consequences of global warming into account, which is not done today, financially feasible becomes a completely different story because so many people are going to suffer so much from the global warming. We have only scratched the surface yet.
Okay, thank you.
Thank you, Professor Meldal. Now, that was Professor Adi Maulana, archaeologist and also lecturer at Hasanuddin University. I would like now to move to high school students.
Yes, students,
Yes,
please
the
ask
future
questions.
generation of scientists over there, Professor. After that, we go at the top. Plenty of time, don't worry. So, can the organizing committee choose high school student there in the corner? Tepuk tangan dulu dong. Warm.
Okay, thank you for the chance. First thing first, I want to introduce myself. My name is Brili Farisaulia from one of the representatives of Attila Islamic High School. I really inspired your career and path to achieve the Nobel Prize. And I know Nobel is not easy to achieve, and I really try to praise it. I have a question on your slide that shows Alzheimer's disease. It's related to protein fibrillations, oxidative stress, and damage to neurons. For a chemistry perspective, how can chemistry help us understand this process and supports the development of long-term, safer, and better treatment for human health? Thank you.
Yeah, so thank you very much for the question. So all of these questions has the answer that we need to have the analytical tools that will allow us to look into this world of chemistry and understand the process of how the proteins are interacting with each other and how we can either inhibit or promote that interaction, how we can do the processing of the molecules, and that requires a lot of analytical tools. So we are using electron microscopy, we are using nuclear magnetic resonance at a high field, we are using high field MS, actually superconducting MS instruments, in order to understand and study the processes in detail. And without this instrumentation, we don't have a mirror, we don't have a looking glass that can see into this world. So that is a very, very important thing, first of all, to have the instruments that is required to do the research. The second thing is to ask the right questions and to look for the answers also where you don't have any answers yet. So that is what we are doing, and I don't think that chemistry is one thing and biology is another thing. I think biology is a small fraction of the chemistry world. So there's no limits between, or there's no barrier between chemistry and biology. It's one thing. We just need to understand the chemistry before we can understand the biology, because the biology is taking the chemistry to a higher level. And it's by understanding the chemistry, it's by studying the chemistry that leads to the biology that we will be able to come up with solutions. Okay?
Okay, thank you.
Thank you. Great question. Tepuk tangan dulu dong untuk adik-adik di sana. At the top, In the balcony, one question. This is another high school student, I think.
Don't be shy, just ask. Yes.
Oh, di sini dulu, Prof. Yang di atas. Sebentar. Ya, silakan.
Hi.
Oh, Master.
Yes, so chemistry. Chemistry is neither good or bad, but humans are. So you can actually select as a human being to use the chemistry tool to do bad things. I don't think that's a good idea. And chemistry is not good or bad at all. Chemistry is actually everything. So you are chemistry. So if you think chemistry is a bad thing, you are a bad thing. Right there.
Thank you.
So I think that chemistry is a very valuable, understanding it is a very valuable tool to come up with solutions to problems. And of course you can also not come up with the solutions to the problems, but just optimizing your profits or whatever you are targeting in your life.
Thank you, Professor. So it is the human behind the chemistry that is probably could be dangerous. So another round of questions. Tadi ada yang di sini, Prof. Harus nadi ya, itu saya nggak bisa lihat. Oke, one, silahkan, mungkin panitia bisa. Women, please. We had
Over
3,
there.
4 men. I would like to have more women asking questions.
Thank you.
Next one.
A very
Yes,
inspirative talk. I'm Rusnadi from agriculture, more on plant physiology. I noticed in one of your slides the role of ROS, which is reactive oxygen species.
Yes.
Plants under stress condition usually deliver many ROS compounds.
We too.
Yes, we too. The question is it possible to use this sort of method, click reaction to increase the to modify the ROS such that the plant can resist to certain stress.
Yeah, so the reason that you get the ROS is the copper 1 that goes to copper 2 with oxygen present. So that condition you can always use to create ROS in plants if you want. That's why cells die when they see copper 1. It's not good for biology to have copper 1. If you have copper 1, you need to have it completely protected by very strong ligands in order to avoid the oxidation with molecular oxygen. So oxygen cannot help to do this. A very low barrier, energy barrier for the oxidation from copper 1 to copper 2. So retrieving an electron by oxygen from the copper 1 is a very, very easy process, making copper 2. And copper 2, in our case, is completely inactive, but in the natural system, the copper 2 is then reduced back by glutathione, for example, to copper 1, and then it can go around this cycle, and all biology is rich in oxygen, so that's a problem.
Thank you.
Thank you, Prof. Rusnadi. Can we have women, please?
We have
Yep.
a woman over here.
Yes, behind Prof. JJ. Yang di sini. And next another woman in that side. Panitia bisa meneruskan mic-nya?
Who has a microphone?
Who has the microphone? Di sini dulu. Tadi sudah di sini. Nanti berikutnya di situ. Oh ya, nggak apa-apa. Di sana dulu. There. So 3 women. Tepuk tangan dulu untuk perempuan.
Yeah.
Thank you for attention that given to me. I'm Sulistiani Jare and I'm from chemistry department, doctoral program in Hasanuddin University. Give applause for chemistry department. Okay. Actually, I have some question. The first one, I saw that your presentation is about copper acid alkyne cycloaddition. Okay. In the periodic table, there are ferrum, cobalt, nickel, copper, and zinc. But you only choose copper. So my question is, why that you choose copper? What is the advantage of Chuprum as atomic center? Okay. For the second one.
One at a time. I'm an old guy, so I would rather have one question at a
Biar dijawab
time.
dulu ya.
Okay.
And let me answer that one. So the copper has very special properties with respect to recognizing pi electrons. So the pi electrons cloud of the alkyne has a very high affinity for copper, and in the crystal structure database, you will find alkynes with up to 5 copper atoms associated with a single alkyne. So there is a high affinity for the alkyne, which is necessary for the pre-organization of the reactants. That's one thing. The other thing is that once you have your reaction, you are attached to the copper atom on the 4 position of the, is that 4? No, the 5 position of the triazole. You have a copper atom still stuck there. It comes off with just a reaction with protonated media, like water. But if it was silver, for example, which also works as a catalyst, it wouldn't leave. So you cannot use it as a catalyst. It will also do the reaction to the triazole, but it will stick to the triazole. So it cannot get off and do another reaction cycle. And if you use the other atoms, they don't work. We have tried everything. The only other one that works to some extent is ruthenium, which forms another isomer of the triazole. So that's actually quite useful. But the copper is very unique in this respect, and we have tried everything.
Okay, thank you, Professor.
Yeah, next one.
Actually, my research is about copper also with a complex compound as an anti-cancer potential. So the second question, may I?
Yeah, last one.
Okay, is copper acid alkyne cycloaddition can switch cisplatin as an anti-cancer potential?
Yeah, I mean, there's many cancer projects in the world today that is based on click chemistry. I don't know if you know the TCO click reaction, which is tetrazine and trans-cyclooctene that can click with each other. In this reaction, you actually have the possibility to release a compound when it is clicked. So you take one of the 2 click components, add it to an antibody that recognizes the cancer, you put it on the cancer cell, and you take the other part of the click reaction and link it up to an anti-cancer drug, so something that kills cells very avidly, and you then let that circulate. It's non-toxic as it is, but when it click on the cancer cell, which has the other component, it will release the anti-cancer drug into the cancer cell. So that's one of the 2 projects that are there. Another one is with the spark reaction, where in particular in diagnostics of cancer, you can get very, very sensitive cancer diagnostics from click chemistry.
Wow. Thank you so much for your questions.
Okay.
Can we give
Okay.
another
Thank you.
opportunity for
Thank you.
another one? So, yep, yang di tengah sini. So, beliau baru saja menjelaskan bagaimana begitu banyak potensi dari click chemistry ini untuk terapi maupun untuk diagnostik.
Thank you for the opportunity, great opportunity that given to me. I'm Umi Zahra from the Chemistry Department UIN Alauddin Makassar.
Yeah.
Okay, Prof, how do you envision the future of chemistry innovation in the area of artificial intelligence and automation? Can your click chemistry integrate it with this technology? Thank you.
Brilliant question.
Yeah, so the click chemistries, not only our click chemistry, but the click chemistries are extremely useful in combinatorial approaches. And combinatorial approaches can be combined with calculations, computer simulations, and also artificial intelligence to select for us the best way to do the combinatorial chemistry, because combinatorial chemistry is extremely powerful but also extremely difficult to carry out. The reason is that instead of synthesizing one compound, we are now synthesizing 500,000 compounds. And how to know where what is and how it's gone, you know, was it a good reaction or bad reaction and so on. With the click chemistries, which gives transformations that are essentially quantitative, we have limited the problems dramatically in the combinatorial research. Also, we know exactly how it's reacting, so we can take that into the computer and use these structures to simulate interactions with proteins. And we can have AI helping us with that. Like, for example, what David Baker is doing. I don't know if you know his work on AI, but he makes AI recognition. So he makes a molecular recognition. I do it by dynamics and mutation, but he does it by AI mutation. So AI is actually figuring out how these two molecules should recognize each other.
Interesting.
Thank you.
Thank you. Panitia di sebelah sana. Tadi ada yang jilbab hitam, kemudian di belakangnya ada next one. Silahkan.
Baik, thank you. Hello.
Yes,
Prof.
hi.
Morgan, nice to meet you here. My name is Nadira. I am from PhD student in junior semester. So I'd like to ask you about what advice would you give for developing research when our result contradict what is commonly reported in the literature? Give some advice for us. Thank you.
So there are two kinds of things. Either it can, we are all experts and we are working very hard to make things work, and then we publish it. And when we publish it, it looks like it's a piece of cake, but it could have taken 2 years for a PhD to actually do it. And that's one thing. The other thing is that I think that the literature, because of the peer review pressure, because of the peer pressure in the research world, I think that there's a lot of results that are half-baked in the literature. So they are not confirmed, and a yield of 80% is maybe only 10% or 20%. So, or that's in a chemical synthesis, or it could be a protein that was actually only expressed in very low amounts, but it's given like you could have several hundred milligrams of it in your expression. So this kind of errors are all over the literature, and that is because we are not regarding this as being important to confirm. And I think this is the key of healthy science, is that you repeat your experiments and do it again and again until you are absolutely convinced yourself that this is correct before you start publishing. So that's one answer. Try to change the mode of publication. The other thing is, as a researcher, don't despair, just do it again. You fail, as I said also in my slide, you will fail many, many times. And you'll just rise and you'll do it again with perseverance and convictions. And if you don't get the results that you expect, maybe you look for what actually happened in your research, because this is where the openings is coming. It's not everything that gives you a new path, but sometimes you are very lucky and your experiment doesn't fall out the way you were expecting it, but something else happens. You discover what it is that is happening and you go with it in your research.
Does that answer your question?
How you maintain your mental?
Resilience.
Yeah, so, okay, I also get this question quite often. You can ask my wife, maybe it's not so good as it looks. But I think that from my perspective, I was always very curious, so I did too many different things at the same time. So I would do instrument building and physical organic chemistry and synthesis and biology and all sorts of things at the same time. So if my stupid synthesis program didn't work, I would just go into the biology and physical chemistry for a while, then I'll turn back with new ideas to the chemistry. So by having different interests and combining these, you can actually travel where you want, actually, where the successes are. And also, you can have one area contributing to the solution of problems in other areas.
Thank you.
Interesting. Thank you so
Thank
much.
you,
So
Prof.
it is about resilience and also being creative. Professor Meldal, I know that you also play guitar and you build your own guitar, right? Can you tell us a bit more about the creativity side and mindful play?
Yeah, I think that science and arts are two sides of the same coin, actually. So if you look at painting, for example, painting is having a concept, an idea, and putting it into 2 dimensions by a 3-dimensional space in the image. So in your mind, you have to picture how to get a 3-dimensional structure into your canvas when you're making a painting, making an animation, whatever you are making that is visual. It's the same we are doing in chemistry. We need to understand in which direction the orbital sticks out that is going to receive the electrons. We need to understand what is the electron density in the different parts of the molecule. And we cannot do that without looking at it inside our heads. So we are doing very much the same as a painter, as a chemist. But I also think that there are other sides that are very important, and one of them is music. Because music is a complete opposite, it's a complete alternative to constructing an image which is more and more complex in your mind. The music is being right here, right now, and in particular, if you're improvising, you get to the next note by listening to what you just played. And this is an important function also in science for your observation of what is going on in a lab. You get rid of your constructed idea, and you are able to actually observe the things that happens right now in your fume hood. So sometimes your textbook is actually full of lies. I know because I'm teaching a very complicated course in organic chemistry at Copenhagen University, and the book that we're using is Carey and Sundberg because that's the best one, but it's so full of mistakes and errors, and the authors have not really been careful in getting rid of all those errors. So you get a lot of half-baked theories or lies out of reading that textbook, but you also get clever at the same time. In particular, if you can pierce through those problems. So that's what I teach my students, to visualize and pierce through the problems in that book.
Thank you. More questions? Yep, Bapak yang di sana dulu, di belakangnya Kaile. Masih ada yang ingin bertanya di sini?
Assalamualaikum warahmatullahi wabarakatuh.
Okay.
Thank you for the opportunity. My name is Birawi Laksono, a lecturer from Medical Faculty, Universitas Negeri Makassar. My question, we're talking about basic science. I love basic science. Before become a lecturer, I work in the laboratory. But previously, I tried to apply for a PhD. Then I was scolded by one of my mentors abroad. She said like this: as a medical professional, you shouldn't do basic science as your main research. You should try to do translational research as a medical student, as a medical professional. So I want to ask, from your perspective, where do we should stand as a medical professional working on the basic science? And if yes, how far? And if we try to work on translational research, how far we can do that? And the second question is,
one question at a time.
Tunggu dulu,
Okay, thank you very much.
nanti beliau lupa.
Yes.
Yeah, please, Professor.
Yes, the translational research or the transformation of existing research into application, and particularly within the field of medicine, is a very, very important task. And it's also a task that can keep you occupied for the rest of your life, essentially. So I think that's a very good target to go for. It's a very good attitude to have. For me personally, I love basic research mainly because of this curiosity and because of the existential component to basic research. So basic research, when you are a basic researcher, you also start ask yourself questions of what is going out there in the neutron stars, what is affecting, what is gravity doing to actually make the large structures that we have out in the universe. How many places do we have Earth-like conditions out there? You know, a lot of philosophical questions, and what can the chemistry do in these places? These kind of questions you learn to think about by doing fundamental research. If you are very occupied with applied research, you might be extremely successful, you might even be financially extremely successful, which is, of course, something we all would like. But for me, it's very important to have the freedom of thought to actually think exactly like I like.
Thank you. Berikutnya, while the mic is there, please, Pak Ilham. Just pass the mic, please. And the next one from here, from this side. Yeah, nanti yang jilbab abu-abu.
Thank you, sister and moderator, for the opportunity. Mr. Professor Meldal, I'm not from chemistry, but I would like to have your view. You mentioned about how our world now is not in a good shape. Everything is now we have to be alert with. I would like to have your view on how chemistry can actually prevent war. You know, I mean, we talk about nuclear war and how this, you know, come into perspective of international leaders of having this. So in terms of chemistry, how the reactions and everything in terms of, you know, weapons, etc. This is maybe out of the context, but I would like to have your view in terms of peace foundations, how we can have that.
So this is not only a peace thing, this is also because there are two things.
Yes.
One thing is that it's
Thank you very much, Nana.
It's not the chemistry itself.
So.
It's the education in chemistry. It's our understanding of the availability of the resources. It's our understanding that if we raise the temperature 2 degrees, everybody should understand that.
Okay.
We double the amount of water in the air. It goes higher up because of the temperature increase. It goes further out, increasing the desert belt, and then it falls down in large amounts out here, creating flooding in Central Europe and US and Pakistan. Right? So this is what is happening, and we only understand that if we are educated in it. So I think chemistry education is a solution. It's also the solution to a lot of the political problems, because we will make the right choices. And I think that's also why it's very important for us to stick to the democracy. We have a beautiful pluralistic democracy in Scandinavia. I think most of this Southeast Asia also have democracies. And then you have 3 very powerful, maybe 4 very powerful autocracies that we should actually be very careful not to get under that umbrella.
Yeah.
Because it's through the democracy process, through the compromises, through the division of wealth, through the division of resources between the populations, that we can have an empathic development of a herd community, which is all that is needed for taking care of the global problems.
If we have this kind of Nobel Laureate like that, we will not have war. Thank you, Nan.
Thank you.
Assalamualaikum.
Next one. Tepuk tangan dulu dong. Sambil menunggu di atas masih ada yang mau bertanya? Oh ya, yang di sana ya. Oke, next one after this
Okay, thank you. But first, let me say hi to Prof JJ. Halo Prof JJ. Kami dari Politeknik Sialan Makassar, kami ada 10 orang, and almost of us is from Hasanuddin University. Jadi
So
sebagian
they are from
dari
outside Unhas.
Yes, but kami adalah alumni dari Unhas seperti itu. Okay, Professor, my major is in public administration, so it's so different with your major. But let me ask you this question. In many countries, we are struggling with our policy implementation, like in science, in economic, in law, etc., between the invention of the science. My question, based on your experience as a scientist and including Nobel laureates, what is your how to influencing the public policy without politicizing the science? For me, I always think that there is a pause time, it's a long time between the invention and the implementation of public policy. So based on your experience, what the government should do? Thank you.
Well, I think that public opinion is extremely important in this regard, and that means that everybody have to talk about it and have to figure out what is actually a stand on these issues, on how to finance the best possible road forward, so that you are electing, because you are still electing, the right politicians that will have that attitude as well. If that is not a main focus, if your main focus is to get bread on your table for the next day, of course that will be what you elect your politicians for, but just keep in your mind that the bread comes maybe a little bit later, but it comes much better if you actually do the education.
I see.
Thank you. So
Thank
you're
you, Professor.
emphasizing evidence-based policy or science-based policy, which is really important. The next one, ada yang di atas? Ya, yang rambutnya panjang. Tuh yang di samping. Ntar.
Okay, good afternoon, Professor.
Good afternoon.
Thank you for the opportunity. My name is Michelle. I'm a nursing student from University of Hasanuddin. My question is, although click chemistry has been applied in biomedical research for many years, but what do you see as the remaining challenges that limit its wider and routine use in clinical practice? Thank you.
So, as I said, there are 5 different kinds of click chemistries out there, and some of them are better than others for some things, and some of them are better for other things. So in clinical research, I think probably the TCO reaction is more important than the actual QAC reaction, because we don't like copper in clinical studies at all. And it's very difficult to get completely rid of your copper molecule, your copper atoms after a synthesis, in particular if this is carried out at large scale for clinical studies. So I think that we should consider what kind of click chemistry we will use in each particular case. And there are chemistries, click chemistries, available to be used in clinical settings. So I think that is actually done already, and we have about 40 to 50 click-based drugs in clinical trials at the moment.
Thank you.
Alright, thank you so much, sir.
Ada lagi yang di atas? Mungkin ini mana Profesor Harman? Last round of questions. Maybe you'll be tired.
What? Yeah, take one more.
Okay. Now this is our last round of questions. So dari atas. More questions. Okay.
Should I choose one?
Yes, yes, I'll let you choose.
The one up there on the stairs. The one up there in the back. Yes, you. Yes, you.
In the back, the very back, yang paling belakang.
That one there. Okay. What? The woman right there on the stairs was very eager.
Ada perempuan tadi yang katanya, siapa tadi yang angkat tangan? Perempuan, cewek.
Yes, that one.
Ya. Sabar ya. Silakan deh.
Good afternoon, sir.
Good afternoon.
My name is Aulia. I'm a second-year student
Hi, Aulia.
majoring in food science and technology at Hasanuddin University, Makassar. So, you mentioned your research is about the click questions. Can click questions be done by everyone? And as you show the presentation, it can be done in the open air. And how does it affect the sustainability? Because usually reaction is being ruined by oxygen. So that's for my questions.
Yeah, so well, you really have to get rid of your oxygen. So what we do is we bubble nitrogen through all our solutions before we combine them. And we make sure that we only have copper 1 there. And we also protect the copper 1 against oxidation by having a ligand that coordinate on 3 sides of the copper, leaving just 1 side for the coordination to the alkyne. In this way, we can do it with catalytic amounts down to 0.005 mole %. So that means that we essentially use nothing to catalyze it if we have the conditions which are oxygen-free. If we have oxygen present, that competes with the catalytic cycle, so then it won't work. But we actually use oxygen-free conditions in all click reactions that we carry out.
Can I ask one more question?
So it's not useful to do it in open air, as many things. Many places it's stated that you can do this reaction in open air. I don't think so.
Okay. Ya lanjut, silakan.
What makes a reaction is qualified as a kick chemistry? As a kick chemistry?
Yeah, I had one slide. I don't know if you remember it, but first of all, you need to be able to carry this out at very low concentrations, so physiological type of concentrations, which means that you have to have a driving force that makes it a quantitative reaction, even at low concentrations. So in the case of the click reaction, this is facilitated by the copper, which is not a single copper atom, but is actually a small copper cluster in the solution. This copper cluster will associate to the alkyne and associate to the acide. So you bring these two in proximity in space, and then they can react with each other. So this is a prerequisite for having a really efficient click reaction. Something that is quantitative reaction-wise, and something that brings the reactants together in space. And then you have several other things, like it should be easy to employ, so easy to install, and there we often use what is called acide transfer reactions, where we can take any amine in any kind of organic molecule, and we can do an acide transfer reaction to convert the amine into an acide. The alkynes come in all sorts of varieties, including amino acids with alkynes in the side chains. You have alkynes just as carboxylic acids that you can attach to amines, like making amide bonds and so on. So this is a very important feature because that actually makes it applicable for so many different molecular structures.
Yeah.
That's answer my question.
Thank you.
Thank you, sir.
Yeah. I just want to ask among these high schoolers, is there anyone want to be a scientist like Professor Meldal?
Up with your hands.
Up with your hands, yang mau menjadi ilmuwan seperti Prof Meldal?
Anybody wants to be a scientist? Yeah.
Okay. 2 last questions.
Yes.
Is that okay?
Yes.
Before we close? Wow, they're really enthusiastic. So you will have photos to later. So ada yang di sini panitia tolong dibantu. Yang paling depan tadi belum bertanya ya? izin ini dosen-dosen Unhas kasih adik-adik ya. Ya silakan adek. Yang paling ujung, baju abu-abu. Terus satu lagi ya sebelum kita close. Ya, panitia tolong dibantu. Yang ini dulu, yang abu-abu. Setelah itu, ya, enggak apa-apa itu dulu. Ya itu deh, yang abu-abu dulu, baru kemudian the guy in. Ya, silakan.
Thank you for opportunity. My name is Hasna Ivania. I'm from SMA Negeri 21 Makassar. And my question, you mentioned that everything that is chemistry. How can early exposure to chemistry help students make responsibility choices for sustainability and guide them toward a future career in research?
To make choices that support sustainability, well, everybody has to make choices that support sustainability. I'm very ashamed to say I still drive a car with a petrol motor. I should get rid of that immediately and get some e-vehicle probably. But everybody in the room here can do things that support sustainability. I think that's a way, if we as a community decide we want to have sustainability, how do we do that? We do it by each and every one of us having the opinion about it and doing the actions that are required in order to get a more sustainable world. It's in our consumption, it's in the way we prepare our meals, it's in the kind of things we are eating, it's in our transport, it's in our clothing, it's in everything around us that we can support a sustainable world.
Yeah, thank you.
Thank you.
One, last one?
Yeah?
Yeah. It's really hard to choose. Okay.
Hello, Professor. Thank you for the opportunity. Thank you for the time, first off. Let me introduce myself. My name is Julian Yahya Tebe from SMA Negeri 9 Makassar, or Senior State High School 9 of Makassar. I'm here to first say the backstory of my question, because as mentioned from the slides, chemistry is very important. It's the basic building blocks of humans, of life itself, the 16 elements mentioned in the first slide. It controls living beings for millions of years, from dinosaurs, our ancestors, even us today. And most of our experiences, whether that is passive, which is our immune system, antibody, even active, like love, is through a chemical reaction. And it has been really intriguing to actually hear about the amazing leaps that has been discovered because of quick chemistry. There has been amazing new developments in pharmaceutical sectors, for example, in medicine and treatments for HIV, Alzheimer's from broken cells repaired because of the quick connecting them, cancer cells, helping antibodies, recognizing cancer cells and killing them, and possibly help with a better immune system. Like, for example, the horse crab that stayed the same for 400,000 years because of quick stabling its innate immune system and its antibody. Because of this, I was really interested on actually the future that could be made when we focus even more in chemistry and more generations contribute in this field to research, because we've heard about the previous questions, risk, benefits, even public perspectives that view chemistry as maybe dangerous, and how chemistry is actually neutral. So I really want to know, from the perspective of Honorable Professor, what could be the ideal way to build the new generation to not only know, but also contribute in this field? Example, maybe chemistry involved in clean energy, in different sectors of creativity maybe, and how even the public could be more aware and have a different mindset and help with developments. Thank you.
Yeah, so I think that it's not necessarily click chemistry that will do it. As I said before, our general understanding of chemistry. And I think that I was asked a question the other day from scientists that now all the low-hanging fruits in science have been picked. What should we do as new scientists? What is the future of science? My answer to that is that it has always been so that the low-hanging fruit were picked. And there are new low-hanging fruit popping up all the time. One of the low-hanging fruits here is, of course, in the new discoveries of energy efficiency, which is circular with sun as an energy source. We have plenty of energy coming from the sun. We just need to concentrate on how can we capture that energy and put it into use for the humanity. The other one, which is a low-hanging fruit, is the molecular recognition. Molecular recognition is a process that is extremely important. It's more important than any other thing we can think of. And this molecular recognition is not understood. Everyone who does simulations and that as a career will postulate to you that, of course, we can calculate molecular recognition, but I'm telling you, they are 1 million factor wrong in many of their calculations. And that is from measuring the affinity of calculated affinities. If you measure them, they are just 1 million times off. We can land something on the moon with a precision of millimeters, but we go 1 million times wrong when we calculate molecular recognition. So there's something that we don't understand there, and I think that there's a huge future for the chemists, the biologists, the physical scientists, who will actually piece that together and figure out what is it that we are missing in our calculations. So that's a low-hanging fruit. There are many other low-hanging fruits out there. We just don't know where they are yet. So that is the problem. How do we do these new discoveries that are new areas that will lead us to a scientific career? And I don't have the answer for that. Just be observant, look for it.
Thank you so much for the inspiring insights from Professor Meldal.
Thank you, Professor.
I know that many of you would like still ask questions, but due to time limitation, we should conclude this. I know this is not the last one. We will meet Professor Meldal. I encourage you to follow his Instagram, and you can also ask questions there. And also he has YouTube, so videos, so you can also learn more. And of course, you can always email him as well. So I hope any of you will follow his footsteps into chemistry and inspire our world and impact our society. Maybe last...
Can I say one last thing?
Yes, please.
Because many, many times you hear this thing that chemistry is old, chemistry is done, there's nothing new from chemistry. I think on the contrary, everything new comes from chemistry, right? In biology, you are essentially just studying the prescriptions of nature. In chemistry, you are actually creative and you are actually discovering new processes, and you are also discovering the underlying mechanisms in biology. So chemistry will never die. It's a wonderful future out there for chemistry.
Wow. Professor, before we close, I would like you to give, if you could give one advice to these young people here and there, what would that be?
Do what you think is right, what you think is most exciting, and I'm sure you will be both honored for it and also recognized for it in the end.
Tepuk tangan dulu untuk Professor Meldal. Now we are coming to the end of this program. So if everything is chemistry, then today we have witnessed a powerful reaction between brilliant mind and eager audience.
Brilliant mind.
May the insight shared today spark a chain reaction of innovations in all of us. Please join me. One final thank you to Professor Morten P Meldal, Nobel Laureate in Chemistry in 2022. Such an honor for me to host you, Professor.
hold that? Wow, it's quite heavy. Is this the original, Professor? I want to take picture. Can anyone take picture with? Thank you so much. Now I leave it to MC. Maybe there will be group picture, Professor. Thank you so much. It's an honor.
Thank you very much, everyone, for coming today. Excellent questions, excellent audience. Thank you very much.
Thank you very much, ladies and gentlemen. You may be seated. Professor Morten, again, thank you very much for inspiring us all today with your insightful knowledge, experiences, and expertise. And to our moderator, Hasnawati Sali, PhD, thank you for leading such a great discussion. Excellencies, distinguished guests, participants, ladies and gentlemen, we have reached the end of this incredible session. And to all the students, we hope this experience encourages you all to keep asking questions and exploring the world of science. On behalf of the organizing committee, we would like to express our sincere gratitude for your presence here today. We hope you leave this venue feeling inspired to make a better world through science. Have a wonderful afternoon, and before closing today's event, let me present you a rhythmic poem. Kuliah di Kampus Unhas, kampus terbaik yang nomor 1. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.